Hi, everybody. Today we're going to continue with our intro to the nervous system. So if you guys look back at our previous lecture, we stopped with the myelin sheath. We have described it both in central nervous system and peripheral nervous system. Myelin sheath was important in terms of providing that insulation needed for the electrical impulse to be rapidly passed through the axon of the neurons, and therefore basically getting a signal from one place to another in a speedy manner. I also want to go over what we identify as nodes of Ranvier. Nodes of Ranvier are basically these gaps that you will see between the myelin sheath. So if you take a look at this picture right here, here is an axon of the first neuron. And basically these structures that are surrounding the axon, those would be your myelin sheath. But notice that there is gaps between each of them. These gaps, as I mentioned, are called nodes of Ranvier. And the importance of these is that it allows the electrical impulse, or what we identify as action potential, we'll discuss it later on, to spread very rapidly down the axon and reach the axon terminal for the purpose of releasing neurotransmitter. Now, because this is such a fast process, it's also referred to as the saltatory conduction. So the channels that are basically associated with creating what we identify as action potential, which the main one being the sodium for the process of depolarization, as you can see. These sodium channels are highly concentrated within these gaps. So when these channels open up, there is such a rush of sodium coming into your cell that creates what we identified as significant depolarization or action potential. Added to that is again the fact that these cells are myelinated. They have that insulation, so you can maintain that positive charge inside the cell until you reach the next node of Ranvier. Notice if that in the bottom picture, if you are lacking myelin sheath on your axon, that a lot of that positive charge that was driven in has been moved out or leaked out as another way, and therefore the action potential will not remain strong. So that's the main difference of having, again, that myelin sheath surrounding your axon to give you that a speedy passage of signaling from the beginning of the axon to the axon terminal. Here is another, again, depiction of the same concept we're describing in terms of myelin sheath and saltatory conduction. The first picture you see is not actually in the axon. If you notice, it is highlighting the region what we identify as the dendrite of your neuron. Dendrites in the cell body have no myelination, no insulation, and therefore you notice that the charge is leaking out pretty fast, and that signal dies down pretty fast too. Now, please do keep in mind that this is known as this type of potential, which we will see in, again, only dendrite and the cell body of your neuron, is actually referred to as the graded potential. And this type of potential is basically happens only in these two locations where there is no myelin sheath. And again, that leakage of charge causes this severe or significant degradation of the signal over a short period of time. The second scenario you see is a non-myelinated axon. So you will have, we're talking about the axon, but again, that insulation is absent. Notice that you will have this stimulation, which causes that basically creating a very strong depolarization or movement of ions. But because of lack of insulation, a lot of these charges will be leaking out, and you can see this, basically the strength of the signal declines. The interesting fact about the non-myelinated axon is that you have channels that are spread throughout the axon, which kind of renew that force of potential, or that potential that is what we identify as action potential. So it is slowing down significantly because of the leakage of ions. However, you also replenish this continuously as you go down the axon. Compared to what we have here is where you have one strong stimulation and then there is no basically addition of new channels to allow that recurring recharging process. The last scenario we have here is what we are actually concerned about, what we will be focusing on for the axon portion is assuming the axon are myelinated. So you will see these basically blue structures around your axon. And notice again, you have the stimulation. You have a strong charge that remains relatively strong when it compared to the picture on the top. And then you can renew that kind of charge that you have within the spaces we identify as nodes of Ranvier. And this process, again, this potential that's created, this electrical impulse that is created by these myelinated axons are much more rapid and therefore called the saltatory conduction. So these are scenarios of what we identify as action potential. And it will make sense as we go through this PowerPoint, what is the difference between what we identify as graded potential versus action potential. Now, to get familiar with some of the terminology associated with your cell, we're going to focus on orthopotential, I should say. We're going to introduce three main vocabulary. You're going to see that and hear it a lot. These words are depolarization, repolarization, and hyperpolarization. So the way I depict this for my students is the following. First of all, the cells that we have in our body always have a negative charge. Now, this negative charge may range from about -70 to close to -90, but they always maintain that negative charge. And we'll go over why that charge is negative as well in a few minutes. But the way I draw this is, if you imagine, here is a chart, here's a graph of basically your regular potential. So if, let's say, my cell has a potential of -70 millivolts to start with. So this is its normal. This is what we identify as resting potential. Your cell is happy, there is no excitation, there is no stimulation. Now, when I start to go, and let's say this is 0 and this is going up is +30 millivolts. So as I start to go farther away from the negative, and I'm going to close to 0 and then going again to the positive aspect, this side, this increase where I'm making my cell more positive, this part is identified as depolarization. Now, eventually, at some point, this depolarization will stop, and you want to start to reverse things back to normal. So as you start to come down from that negative charge back to that, in this scenario, that -70, you're making the inside of the cell more negative, and therefore, we're going to use the word repolarization, as in you're returning things back to normal, if that helps you. Now, what if I'm at -70 and I still continue to go farther down? So I'm basically, what we're doing is going even farther than that normal range of -70. So going and dipping below -70, that's identified as hyperpolarization. You're basically going above what is acceptable, becoming excessively negative. Now, please do keep in mind that regardless of if you're depolarizing, repolarizing, or hyperpolarizing, your cell goal is always to go back to that resting potential. In this scenario is -70. So these three keywords are extremely important for your understanding of both graded potential and action potential. Now, another aspect that I want to take a look at is looking at the type of channels that are present, giving me the capacity to either create and maintain my resting membrane potential. As I mentioned, typically about 70 up to close to negative 90. Channels that are involved in the process of depolarization, making my cell more positive, repolarization, making my cell more negative, and then hyperpolarization for an excessive negative scenarios. So these channels are categorized as follows. What we identify as sodium and potassium ATPase. Now we're going to go over this in just a few minutes as well, but just kind of highlighting this whole ATPase. That means that this channel requires ATP to properly work, which makes this process an active transport. And the word active transport implies that you're moving something against the, well, active transport implies that you need energy, ATP, or some other form of energy. But in this scenario also implies that the ions, sodium and potassium, are going to be moving against their concentration gradient, which basically means you are forcing molecules, or ions in this case, to go from a low concentration to a higher concentration. We also have a category of channels called leakage channels. Leakage channels work by the means of diffusion, or they rely on diffusion. They basically move from high concentration to low concentration, either into the cell or out of the cell. That depends on their concentration. But anytime you guys hear the word leakage, that implies the process is a passive transport. And passive transport implies that there is no energy input for that movement. You also have channels identified as voltage-gated channels, which we shorthand to VGC. There are three categories that I want to discuss with you guys. One that are called sodium VGC, ones that are called potassium VGCs, and ones that are called calcium VGC. These channels, as the name implies, will be normally closed or open, depending on the scenario, and will undergo changes when there is a change in voltage or electricity. So remember those numbers that I gave you, that negative 70, positive 30, these are numbers that can literally influence the voltage-gated channels. And again, I'll go into details about when these channels actually open or become active, when these channels become inactive, where are they present within your neuron, and are they involved in action potential or degraded potential. We have channels called chemically gated channels or ligand gated channels. These are channels that are typically corresponds to neurotransmitters. So ligand or chemically gated channels are the same. And again, they correspond to the presence of a specific molecule. For our concern, these molecules are going to be your neurotransmitters, which causes the opening of these channels. Mechanically gated channels, as the name implies, they are sensitive to fluctuation in terms of pressure. And again, I'll go into more detail about them. These are not the ones that are necessarily something we'll discuss for this class, but it is good to know that the receptors, for instance, that are present in your skin surface are identified as mechanically gated channels. For them to be activated, there should be some sort of pressure or force or a stimulation of some sort to open these channels to create an electrical impulse. So let's go ahead and get started with these categories of channels in a little bit more detail. First type we have are identified as your sodium potassium ATPase. As you will go over this, these are extremely important in helping you create as well as maintain what we identify as electrochemical gradient. Now, if you guys recall, a normal cell, and this is something that I have discussed previously, but just kind of going over it again. When you look at your cell, what we have here, you have your extracellular fluid, and inside your cell, you have intracellular fluid. Now, within this fluid, of course, you have water, different water, and then you have a lot of ions and proteins involved in this. Now, what we're concerned about is the concentrations of these ions. So the main ions we're focusing on are sodium, potassium, calcium, and chloride. Now, these ions are also found inside the cell. So you will have sodium, potassium, calcium, and chloride. So when you look at the concentrations of these ions on either side of your membrane, sodium is going to be high outside, making it low inside. Potassium is low outside and therefore is high inside the cell. Calcium is high outside and low inside. And same applies to your chloride. So why am I drawing this for you guys? When we're talking about the channels, such as your leakage channels, your voltage-gated channels, or these channels, or even your ligand-gated channels, whenever they move ions, they move it based on their concentration gradient and via the means of diffusion. Which means if I'm using, and remember this, guys, if I'm using these type of channels, which is your leakage, my leakage channels, my voltage gated channels, and then my ligand channels, if these channels are open, they're always going to move ions with their concentration gradient, which means they are going to move ions from high concentration to low concentration. On the other hand, we have your sodium potassium ATPase, which are going to move ions against their concentration gradient. So if you look at this scenario, sodium is high outside and sodium is low inside. But what is this channel going to do? Is going to take 3 sodium from inside the cell and do what? Pushes it outside. So 3 sodium in this scenario is gonna move outside against its concentration gradient from low to high. On the other hand, while this is happening, you're also taking 2 potassium from a low concentration and bringing that inside the cell. So again, you're moving it against their concentration gradient. This type of movement, again, because it's opposite of their original direction or concentration gradient, they require energy input. In this format, the energy input is ATP. And the process is, again, active transport. Now, again, when we're talking about leakage channels, both sodium leakage and potassium leakage, ions are going to be moving from high concentration to low concentration, which means if you're looking at this scenario, this is your ECF or outside, and this is your ICF. And you notice that potassium is leaking out from high concentration to low concentration, and sodium is moving from high concentration to low concentration. Again, moving with concentration gradient does not require any form of energy in terms of cell input, and therefore is identified as a passive process. Now, the next thing we have is your voltage-gated channels. So there are 3 types of voltage-gated channels that I've listed for you: potassium, calcium, and sodium. So in this scenario, we're talking about calcium and potassium. These channels are basically either staying closed or open. So open or closed, pretty simple. So logically, again, kind of sticking to that whole idea of the ions, and I'm gonna redraw this for you guys until everybody kind of becomes second nature for them to do this when we start a concept in terms of ion movement. Potassium is high inside while calcium is low, calcium is high outside. So if I have voltage gated channels, again, these channels are sensitive to voltage, so they are opening or closing at very specific voltage or electrical impulse. When your potassium voltage gated channels open, potassium voltage gated channels are open, they are going to allow the potassium to move from inside to outside, again, from high concentration to low concentration. On the other hand, if we're talking about your potassium, when we're talking about your calcium, the calcium is going to move from outside into the cell. So the direction is opposite. Your focus always going to be where is the ion high and where is the ion low. And for these type of channels, the ion's always going to be moved from high concentration to low concentration. And these channels, these are specific ones we're talking about, they are opening and closing with a specific voltage. I want to kind of show you this also. So here is my channel. If you look at the picture, these things you see at the bottom. So these are the gates basically right here and right here. So the gates are now, sorry, I'm just going to draw this a little bit bigger so they can come together. So right now, these gates are closed, no ions can pass. Notice the gates are now going into open position, which allows the ions to move through. So what is so particular about the last type of voltage gated channels we have? And that is that they have three stages actually. One is closed, the other one is open, and one is inactive. And you'll learn about why that is important later on as well. So right now, what we're gonna understand is that for sodium voltage gated channels, you actually have two sets of gates. The first gate is very similar to what we described in terms of potassium. So these are these gates right here. You see them in an angle. These are called activation gate. And you have a second gate, which is this little thing you see here hanging, this one. This is your inactivation gate. As in when this gate is engaged, when it's in place, then your channel would be inactive. But when the first gate is closed, then your channel is literally closed. So let's look at this in a picture. So right here, if I say, what is your closed stage looks like? You have your activation closed, so no sodium is gonna come in, but your inactivation gate is just kind of floating in there. It's not really doing anything. So if I say which of the two gates are actually closed, when you're talking about the closed phase, your answer should be the activation gate. Now, there is a voltage change, and as a result, your activation gate, which is this one right here, is going to come to an open position. So now we have the activation gate open, and again, the inactivation gate is just hanging in there. It's not really doing anything. Now the path is open for sodium to go from high concentration, which is outside, to the low concentration of sodium inside the cell. Now, eventually you reach another voltage, and during that voltage change, you will not close your activation gate. So look, your activation gate are still in an open position. What you will be closing is you place in your inactivation gate and block the channel. So at that phase, you call that the inactive phase. So one more time, activation gate is closed, inactivation gate is open. We identify this as a closed channel. Both gates are open. We call that an open sodium voltage gated channel. Your inactivation gate is closed, but your activation gate is wide open. That is called the inactive phase. And I will discuss this in detail, why that is such an important, because it creates something we call absolute versus refractory period, and ensures that the electrical impulses only moves in one direction when it is sent down the axon. And again, I'll go into more detail about this later on. Chemically or ligand-gated channels. These channels, as I mentioned, are going to be sensitive to typical molecules, or should I say typical, sorry, specific molecules. In our scenario, those molecules would be your neurotransmitters. Now, the gate that are described here, and this is the one that we will be focusing on, is your sodium-potassium channel. So look at this scenario. Again, you have your plasma membrane right here. You have sodium and potassium and calcium inside and outside the cell. And you have a neurotransmitter, acetylcholine, ACH, that is present in an environment. Now, as soon as this ACH binds to its receptor side, which is these little cavities there, that as soon as it binds, this channel goes into an open position. In such a scenario, you are going to move your potassium out and your sodium in. Now, this channel, they do not, I don't know why they draw a calcium here. This specific channel should not allow calcium to move in as much, even though a small amount might move in. The main goal of this channel would be to move your sodium and your potassium on in or out of the cell, depending on their concentration gradient. Now, the other two that are not listed here, but you also need to know, is potassium channels. Again, these are ligand-gated channels. Now, use your logic again. If I draw this again, calcium is going to be what? Sorry, I keep saying calcium. Potassium is going to be high inside the cell, right? And low outside the cell. So if I have a ligand-gated channel, okay, and normally these are closed, as soon as it opens, it's going to push the potassium out of the cell with its concentration gradient. Now, if you're unfamiliar with the word efflux, efflux literally means that moving something out of the cell. So in this case, you're effluxing your potassium. Chloride, on the other hand, if I'm again looking at the concentration gradients of chloride, chloride is going to be high outside and chloride is going to be low inside the cell. Therefore, when these channels open up, chloride is going to move from outside to inside, or from high to low. And this is the word that we use to describe this concept, is influx, as in molecules are moving into your cell. Last type of channels that we have are called mechanically gated channels. And again, these are typically ones that are correspond to some sort of physical changes or physical distortion. They can be sensitive to your sense of touch, pressure, or vibration changes. So notice this channel right now is closed. And as soon as this channel feels some sort of pressure changes or vibration, the channels open up, causing ion movement, and that creates the basically electrical impulse for the activation of the neuron. Now, the next topic we're going to talk about, and then we'll move on to the actual neuron in a little bit more detail, is the concept of excitable cells. So excitable cells are basically cells that are going to be responding to changes in their environment, and the changes in these environments are electrical signals. So basically, if you have a cell that is excitable cell, when they see or are stimulated by an electrical impulse, they get activated or they get quote unquote excited. The category of cells that are identified as excitable cells in our bodies are neurons, muscles like your skeletal muscle, your heart, or your smooth muscle, and your secretory cells such as your glands like pituitary gland, secretion of insulin from your pancreas, etc. Now, we won't talk about pancreas or pituitary gland in our course. You will see that in more detail when we talk about endocrine system lecture. Now, for us to create what we identify as an excitable cell, the first thing you want to do is create a separation between the charges as well as the concentration gradient. So basically, when I'm moving ions and placing them with different charges on either side of my membrane, I create what we identify as electrochemical gradient. Electrical implies that we're talking about differences in charge, and chemical, we're talking about concentration gradient. Now, as soon as I do this kind of shift, as soon as I have this variation in gradient, I create what we identify as potential energy. Basically, I have some stored energy as I move these particles in either side, as I see a separation of the charge. So as soon as there is a mechanism that allows these to move, then these ions can move either in or out of the cell and create what we identify as excitation. Now, electrochemical gradient, again, is described as sum or addition of the electrical and chemical forces that are basically working on your plasma membrane, your phospholipid bilayer. Again, this is acting as potential energy. What are some of the factors that allow us to create this? We can have charges like negative proteins inside the cell. That's another contribution. We can have a really high concentration of potassium inside the cell. And then on the other hand, on the other side of our membrane, in our extracellular fluid, we will create a high concentration of both sodium and potassium, which is your typical salt, basically, Na and Cl. Now, again, looking at this at a little more detail, electrical gradient, what is that exactly described? It is the amount or concentration of positive as well as negative charges that are found across your membrane. So always remember, if you have a positive and negative, they're gonna be attracted to each other, right? Opposite attract, kind of think about that. So anytime you have a lot of negative in an environment, which is typically inside your cell, a lot of positive ions want to come into your cell, which in this case, for our body, that's sodium. So if sodium has a chance to get into your cell, it's gonna rush as fast as it can into your cell, so it can basically eliminate that gradient in terms of electrical gradient. Chemical gradient is a variation of ion concentration, as I mentioned, right? So we have high sodium outside, right? Or in your ECF. We have high potassium in your cell or your ICF, right? And again, anytime you have that gradient of the ion, your ions want to diffuse down their concentration gradient. Sodium wants to move from high to low, which means sodium wants to come inside. Potassium, if it has a mechanism to get out, it wants to again go from high to low, which means from outside inside to the inside of your cell. So when we're talking about electrochemical gradient, is a twofold process, right? So we're talking about one aspect that positive and negative are attracted to each other. So if you have a negative, a high amount of negative in one place, positive would be attracted to it. And then if you have an imbalance on the concentration of your ions, you also will be seeing that shift or movement of ions from high to low. So imagine you have a cell. This is baby cell, it's just born. And at that point, you really don't have any electrochemical gradients. What does that mean? Is that you basically have a cell, and this is not something you can visualize or you can see inside the cell. This process happens very rapidly. So you have a cell, and that cell is basically born, and I put that in quote unquote, as equal concentration of sodium and potassium inside the cell and outside the cell. So when you have a cell, here's my cell. The two main ions we have is sodium, which is equal inside and outside. Oh, sorry, this should be potassium. And then your second ion again will be potassium. You have equal amount of it inside and outside the cell. Now, what allows me to move more sodium outside and create a higher concentration of sodium outside the cell and higher concentration of potassium inside the cell would be one type of channels. And those are your sodium potassium ATPase, which basically going to use the energy of ATP to move 3 sodium out of the cell and 2 potassium into the cell. So to kind of help you visualize this again, this is your initial cell. I'm going to draw this a little bit bigger so I can label it. And you will have in your membrane, and I'm only drawing one, but remember you will have many of these channels. And this channel is your sodium potassium ATPase. What is this channel going to do? It's going to take 3 sodium from inside the cell and push it into the outside. At the same time, while this channel is working, it's going to take 2 potassium from the outside environment or from your extracellular fluid and bring it into your cell. So when you start with equal numbers, now more sodium is being pushed outside and more potassium being pushed inside the cell. So what you eventually gonna end up with is low sodium inside the cell and high potassium inside the cell, right? So if you do this, you're establishing what? You're establishing electrochemical gradient. You have more positive outside the cell, right? And you also have a concentration gradient for both your sodium and potassium. Now, this is only one channel. How can I maintain this newly established charge of, or newly established electrochemical gradient? So first of all, your channel is going to continue working. So it's not like because it's already established this concentration gradient, it's going to stop working. No, this channel is always working, continuously pushing 3 sodium out and 2 potassium in. But eventually, what happens is you're going to end up with an imbalance, right? So you're going to have basically pushing all of your sodium out of the cell and all of your potassium into the cell. So there wouldn't be any sodium inside and any potassium outside the cell. So how do I maintain the electrical chemical gradient that I've originally established? How do I maintain a higher sodium outside the cell and lower inside and higher potassium inside the cell? How do I maintain that? We're gonna go back to your leakage channels. So if you think about your same picture that I have here, so I'm just gonna add to that. So in this side, or around your cell, not just on this side, you have what we identify as your sodium leakage channel, okay? So sodium leakage channels. Now remember, you have a lot of sodium outside. So what is this sodium wants to do? Sodium wants to come into the cell, right? From high concentration, which is around the cell, into your cell. Now, as sodium is coming in, what is happening to those sodiums? They are eventually getting pumped out of the cell, 3 at a time with your sodium potassium ATPase. So basically, you have sodium coming in through leakage channel and then being pushed out of the cell through your sodium potassium ATPase. And they are helping each other to maintain your electrochemical gradient for your sodium. You also have your second category of your leakage channels, and those are identified as your potassium leakage channels. So potassium channels, I'm gonna draw multiple of these, are actually more common in your plasma membrane. And they are going to allow lots and lots of potassium to what? To leak out, right? So membrane is more permeable to them. So more potassium is gonna leak out, right? So you're only bringing 2 potassium in, but a lot of that potassium is being leaked out pretty fast. Why is that important? Why are we talking about this in terms of sodium and potassium and how effectively your leakage channels are doing their job? Think about the charges that I told you associated with your cell. We said that your cell, when it is at rest or just maintaining its electrochemical gradient, your charge would be always negative. So if I'm taking a lot of positive out of my cell by pushing a lot of potassium out, but only replacing a small amount of that, then what is the tendency of your cell? The tendency of your cell would be to become more negative because you're pushing a lot of positive out of the cell, okay? Hopefully that makes sense. So pay attention to the questions when we're talking about establishment versus maintaining. When you originally establish it, the only channel that is involved is your sodium potassium ATPase. After the establishment, if I want to basically continue keeping that electrochemical gradient when your cell is resting, so it's not excited, it's just resting, I need the involvement of both the potassium and sodium leakage channels, potassium being more effective in moving potassium out. And at the same time, I also need the sodium potassium ATPase to basically push ions to where they came from so your leakage channels can continue doing what they were doing. I'm going to stop here. We're going to continue looking at the variation between resting, graded, and action potential, which are involved in the excitable cells or neurons for the nervous system.